Thank you, Angus. Good morning, everyone. My topic today is fruit set and crop lot management of Australian-bred scab-resistant apples. Before I start my talk, I would like to tell my audience that my talk will be in two sections. First, I'm going to talk about a pollination experiment, followed by a hand thinning experiment. So please don't confuse if I started talking about something very different. So this is the first section of my talk, and it is a pollination experiment on Kaylee. So Kaylee is the first scab-resistant apple developed from Appleton Research Facility breeding program. It matures 2 weeks before pink lady. Kaylee is a really sweet, low acid, crispy, juicy, with three weeks shelf life, and it has got a semi-spur growth habit. As Kaylee is newly developed, several aspects of its physiology are largely unknown, like its pollinizer varieties, or we're not even sure or doesn't know whether it's self-fertile or not. Therefore, the objective of this study was to see the efficiency of different apple varieties as potential pollinizer for Kaylee, and we're also interested to check its self-fertility. So what is self-incompatibility? Well, self-incompatibility is the mechanism that allows the pistil of a flowering plant to distinguish between genetically related and unrelated pollen to prevent inbreeding and promote outcrossing. In apple, incompatibility is mainly caused by a gametophytic self-incompatibility system due to the presence of a gene called S locus. So in apple, for a cultivar to be compatible, the S locus gene of male flower and the female flower has to be totally different. So in this picture, you can see, in the first picture, pollen is rejected. That's because the S locus gene from the pollen is identical to that of the female part. And on the second picture, pollen is accepted. That's because the pollen grains, whether you do it through artificial pollination or through bees, is completely different from that of the female part. That's why the pollen is accepted, and there will be fertilization. So this is a flowering time of different apple varieties grown in our research station, and these are some of the varieties I have used as pollinizer for Kaylee in this experiment. So in this graph, as you can see, there are two different varieties, different groups. One is the early varieties, and the second is the later varieties. So this red dotted line is the Kaylee, and the varieties, pollinizer varieties are used were Manchurian, Braven, Sandioner, Pink Lady, and then I also use Kaylee on Kaylee. And there is another variety also I use, which is RS103-110, which I'm going to talk about later. So, and it's obvious from this graph why I use these varieties. It's because of the synchronization effect, and the later varieties, it was too late, so I couldn't use them. So we have carried out emasculations and artificial pollination experiment at balloon stage, and it was September last year. And after 1.5 months later, we have counted the fruit set. This is that's a Kaylee tree, and these different color ribbons are the different apple varieties I have used as pollinizer. Okay, so some of results from these pollination experiments. Based on this table, I have categorized those varieties into 3 groups as high, medium, and low. So percentage fruit set, Braven, 110, Sandioner. They fall into high categories, followed by Pink Lady and Manchurian. And Kaylee and Control, they have very poor percentage fruit set. And the seed number also, Braven, 110, Sandioner, Pink Lady, they have got good number of seeds, followed by Manchurian and Kaylee, and control, they have low number of seeds. So in conclusion, I would like to say that appropriate pollinizer varieties for Kaylee could be any of these: Pink Lady, Manchurian, Braven, Sandioner. That is, there is no any, you have got plenty of choices if someone wants to grow Kaylee. So you don't have to worry about pollinizer varieties, there's plenty in there. And we also found that Kaylee is self-incompatible. So ladies and gentlemen, this is my second part of my talk, and it is on a hand thinning experiment carried out on 1/10. And this is the second selection from our ARF breeding program. And it's quite a early variety. It matures immediately after Royal Gala. And like Kaylee, 110 is also a sweet, crispy, juicy apple tolerant to alternaria. However, fruit of 110 tends to be quite small to medium, so fruit weight is 130 to 145 grams. And this fruit size can limit orchard productivity. Therefore, the objective of this experiment was to determine the latest timing at which crop loads should be established to maximize fruit size and yield. So this hand thinning experiment was carried out on 10 years old 110 trees at Appleton Research Facility. They were grown on rootstocks MM106, 12, M26, M9, and Ottawa. And the trees were hand thinned during November to December. And the thinning timings were 5, 6.5, 8, and 9.5 weeks after full bloom. So some of the results from this experiment shows that thinning at 9.5 weeks after full bloom reduced average fruit weight by 10 grams relative to 5 weeks after full bloom. And also, thinning at 9.5 weeks after full bloom reduced average fake out of first grade fruit to 83% compared to 92% achieved by 5 weeks after full bloom. And this reduction in first grade fruit is primarily due to the increase in production of smaller apples at later pruning time. So this is a graph explaining what I have just explained in the previous slide, effect of timing on mean fruit weight and on first grade fruit at harvest. So on the vertical axis is your fruit weight in grams and percentage fruit, first grade fruit, and on the horizontal axis is your thinning time. So here you can see that each week the thinning timing is delayed. You can see a steady decline in fresh fruit weight in grams, and also a decline in first grade fruit pack out percentage. So I'd like to conclude here that in order to maximize your fruit size and yield of 110 apple, fruitlet thinning should occur as early as possible after flowering, and don't delay after that. So that's it. Thank you.